Okay, go ahead.
Hello, my name is Zihan and I researched the TRPM4 and TRPM5 with my mentor, Professor Catherine Melder. So a little background information, TRPM4 stands for transient receptor potential melastatin submember 4, and TRPM5 stands for transient receptor potential melastatin submember 5. So throughout this research, I'll be mentioning TRPM4 KO, which stands for TRPM4 knockout, which means that the cell does not have TRPM4, and vice versa with the TRPM5 knockout. And I'll also be mentioning TRPM4/5 DKO, which stands for double knockout, which means the cell does not have both TRPM4 and TRPM5. So TRPM4 and TRPM5 can be found in tissues and cells. They can be such as mast cells, neurons, etc. But I'll be specifically focusing on the taste signaling transduction context. Taste cells are found in four different types of cells. Type 1 glial cells, which are support cells. Type 2 cells, which handle bitter, sweet, umami taste stimuli, which is the left one on the picture. And type 3, which stands for sour and salty stimuli, and then BR cells, which stands for broadly responsive cells, and it handles bitter, sweet, umami, and sour taste stimuli. So in taste cells, the activation of G protein coupled receptors stimulate the PLC beta, which produces the IP3 and then binds to IP3 receptors to trigger calcium release from internal stores. Both TRPM4 and TRPM5 are activated by intracellular calcium. However, they're not permeable to it and only allow smaller non-valence cations like sodium and potassium to pass through. So my research question is, to what extent are TRPM4 and TRPM5 similar and different in their roles in taste signal context? Because they're very similar as they both handle bitter, sweet, and umami taste stimuli, I wanted to focus on how they're different and similar and how they work in taste signaling transduction. My method of the research is a literature review to connect all of the existing information. So rather than experimenting with new concepts, I wanted to draw conclusions on existing data. And I did this by utilizing scholarly databases and peer-reviewed academic journals such as Google Scholar and ScienceDirect. For my first source that gave me background information on TRPM4, it taught me that 9-fentanyl stop the flow of sodium ions. So it is a TRPM4 inhibitor. So it prevented the responses to bittersweet umami taste stimuli. Specifically, the ability to detect sour stimuli was reduced. This confirms that TRPM4's role in detecting bitter, sweet, and umami stimuli, as well as its responsibility for sodium entry in broadly responsive cells. TRPM4 played a bigger role in detecting bitter, sweet, umami stimuli in BR cells compared to TRPM5. And for the second source, it taught me about the background information for TRPM5. So it consisted of 24 axons on chromosome 11 and harbors an open reading frame of almost 3,500 base pairs. And TRPM5 is highly related to TRPM4 in homology as it has a 40% similarity at the amino acid level. And in mice, PLC beta 2 and TRPM4 are crucial for tasting bittersweet and umami taste stimuli because lacking either of these components hinder the sensitivity to these tastes, but maintains the ability to detect sour and salty stimuli. So what I drew from my research for the similarity is that they have a 71% of TRPM4 cells are expressed in TRPM5 cells tagged with green fluorescent protein, and 92% of TRPM5 GFP cells are expressed in TRPM4. So if you look at the picture on the right, they overlap. So that is what the GFP stands for. They're overlapping by 71% for TRPM4 and then 92% for TRPM5. And mice without TRPM4 and TRPM5 did not react to taste stimuli, which shows that both TRPM4 and TRPM5 are needed in order for the mice to taste bittersweet and umami taste stimuli. And TRPM4 and TRPM5 work together to complete taste signal transduction, which was the conclusion drawn from my research. The differences between TRPM4 and TRPM5 is the different concentrations to which they're sensitive to. So TRPM4 is specifically blocked by intracellular ATP and adenine nucleotides, while TRPM5 is insensitive to it. And TRPM4 is insensitive to acidic pH, whereas TRPM5 is blocked by acidic external pH of 6 or lower. They also have different sensitivity to intracellular calcium. So TRPM5 is more sensitive in excised patch recordings, needing about 5 to 10 times less calcium to be activated. And even after desensitization, it is still about 6 times more sensitive. The implications of my research is in future research, they should study how to regenerate taste cells because as people age, our taste cells deplete, and when they deplete, we lose our appetite, and oftentimes elderly people um, they lack nutrition because they don't want to eat food. So regenerating taste cells and manipulating taste cells can um, make people have more appetite and help the elderly with eating better and like have flavor enhancement. The next topic of research that they can do is manipulate TRPM4, and TRPM5 to enhance stimulus perception. So instead of manipulating the taste cells, they can develop drugs or foods that activate these signaling pathways to provide flavor enhancement. Some limitations of my study is that my study is solely based on the data of other people's study, and I have came across some studies that have inaccurate data, so I had to be like careful with the research articles that I was reading. And also some research is more than 10 years old, which may affect the credibility of the data that was collected. These are my references. And I would like to say a special thank you to my professor, Catherine Melder, because I had a lot of meetings with her. It was very informative. She answered all of my questions, and I was very curious about a lot of things. And she was really understanding of that, even though like she was a professional in her field and I didn't understand anything. And there was also fast communication, like we emailed a lot, and she basically like emailed me back on either the same day or like the next day. So like whenever I had questions, it was very easy to get answers and help from her. Thank you.